So today I'll discuss on enzyme kinetics. Enzyme kinetics is basically the study of chemical reaction that are catalyzed by the enzyme. So the study of enzyme reaction rates and how they change in response to changes in experimental parameter is known as kinetics. One of the key factors affecting the enzyme reaction is the concentration of substrate. So here is a graph. It is known as the Michaelis-Menten graph, which states the relationship between the substrate concentration and the rate of reaction. That is denoted by V. So in this graph, what we observe here, like initially, once we raise the concentration of substrate, so there is a fast increase in the rate of reaction. But at some point, even after increasing the substrate concentration, there is no change in the velocity, and it has acquired the Vmax condition, means the maximum velocity it has acquired, the system has acquired. And the reason is that because in this particular system, we have kept the enzyme concentration constant. So once all the enzymes at this level, all the enzymes are being saturated or engaged with the substrate molecules, so there is no more enzyme available in the system. So even after increasing the substrate concentration, there is no rise in the velocity. That is the reason, and that's how we get the plateau. And this graph we get, that is known as the hyperbolic rectangular graph. And what are these terms? The V0 is the initial velocity, while the Vmax is the maximum velocity, and Km, what is Km? Km is also known as Michaelis-Menten constant. It is basically the substrate concentration which is being expressed in moles per liter at half maximum velocity. Means that concentration at which the system can acquire the half maximum velocity. So that is the Michaelis-Menten constant, and S is the substrate concentration. So what is the significance of this Km or the Michaelis constant? The Km provides the amount of a substrate required for significant catalysis to occur. It is the measure of affinity of the enzyme for its substrate. A high Km indicates the weak binding and a low Km indicates the strong binding. So higher the Km, lower the affinity, and the lower the Km, lower the affinity, higher is the Km. Now, what is the significance of Vmax? What information we can infer from the Vmax? The Vmax of a reaction is an index of catalytic efficiency of an enzyme. Rather, the Vmax by Km is more reliable. This ratio is more reliable indicator of catalytic efficiency of any of the enzyme. So Vmax is useful in comparing the activity of one enzyme with that of another. And now this is another plot that is known as line Weaver-Berg plot. So what is the importance of this particular plot, and what is why it came into the existence? So that is also known as the double reciprocal plot. Sometimes it becomes very difficult or impractical to achieve the high concentration of the substrate to reach the maximum velocity. So graphically it can't be shown from the Michaelis-Menten graph. So the experimental data at lower concentration is then plotted as a reciprocal. So all these values like V, instead of V we have taken 1 by V0 here, and here the substrate concentration we have taken as in reciprocal. So this states like that's how, because two major variables have been reciprocate, that's how this graph is known as double reciprocal graph. And this slope is the value of Km by Vmax, and that is an indicator of catalytic efficiency, and this is the -1 upon Km values. Enzyme inhibition, any substance that can diminish the velocity of enzyme reaction is called an inhibitor. So, the two classes of inhibitors are, one is the reversible inhibitor, another is the irreversible inhibitors. Inhibitors can wind up to the enzyme via non-covalent or covalent interactions. Mostly in the irreversible inhibition, there is a covalent binding of the inhibitor to the enzyme. While in the reversible, it is like interaction of the inhibitor to the enzyme via the non-covalent interactions in the form of hydrogen bonding, ionic bond, or in the form of hydrophobic bonds or weak bond of wall forces. So this is the classification of enzyme inhibitors, reversible and irreversible. Reversible again can be of three types: competitive inhibitors, non-competitive inhibitors, and uncompetitive inhibitor. And while in the irreversible, there can be three categories. One is the reactive substrate analog or affinity labels, then group specific irreversible inhibition, then suicidal inhibition. We'll discuss one by one. So this is the diagram of the competitive inhibition. So this is the enzyme. It has some active site, and this is the normal substrate which can better fit in this particular site. And this is the normal trend, how the substrate interact with the enzymes and leading to the formation of enzyme substrate complex. And after being breakdown, it converts up into the product. This is the normal pathway. So this is the inhibitor, which is like a structural analog of this substrate, actual substrate. So there is a competition between the two, and who can like better no wind up with the active side. There is a competition between the two. So this is how, like, instead of, if the concentration of inhibitor is more as compared to the substrate, so it will predominantly wind up with the active site and block the entry of the substrate, and thereby no formation of the product. So, but if the concentration of substrate has been increased, so this competitive inhibition can be arrested to some extent. So, inhibition could be overcome by increasing the substrate concentration. So this is actually both are structural analog and both have equal, opportunity to wind up to the active site. So it is the concentration dependent which is going to wind up first. Now, if we look at the Lineweaver work plot, the reciprocal plot, so we will see the two important feature in this particular graph. One is this 1 upon V max, it remains unaltered. The green line shows the graph when there is no inhibitor. It is a straight line graph, and wherever it cuts onto the x-axis, this is that indicates the Km. One negative of reciprocal of Km. So while in the presence of competitive inhibition, there is a increase in the Km value. The Km is now increased, means affinity is lost. Affinity of enzyme with the actual substrate is diminishing, and while the V max remains unchanged. So two important feature in the Lineweaver work plot. The enzyme kinetics of the competitive inhibitor is that the V max is unaltered while the Km is increased. So just by seeing and look at this graph, we can immediately notice whether this is the graph stating the competitive inhibition. Now here are few examples showing the competitive inhibition. One important example is sulfonamide. It is an structural analog of para-aminobenzoic acid, and it inhibits the synthesis of folic acid in the microorganism. And that's how they act as an microbial agent. Isoniazid, it is an anti-tubercular drug, inhibits the biosynthesis of NAD and restricts the growth of organism and that cause the tuberculosis. Another example can be the dicambrol. It is an anticoagulant drug, is structurally similar to vitamin K, and it inhibits the vitamin K activity and inhibits the formation of prothrombin. Then physostigmine, it inhibits the acetylcholinesterase, and it's used to treat the glaucoma and myasthenia gravis because it is a competitive inhibitor of acetylcholine. Then move on to the non-competitive inhibitors. So what is the important feature of these inhibitors? The non-competitive inhibition occurs between the substrate and the inhibitors, and inhibitors is usually structurally different from the substrate. So what we have seen in case of competitive inhibitor, there was a competition between the actual substrate and the inhibitor. But while there is no competition between the actual substrate and inhibitors, because inhibitor is actually is structurally different from the substrate, and it also bind at the site on the enzyme molecule other than the substrate binding site. So we will see in the diagram ahead how they are not no, both are not competing with the same site for binding to the enzyme. Then non-competitive inhibitors bind free enzyme or enzyme substrate complex. So either way, the non-competitive inhibitors can bond to the free enzyme, or they can bind to the enzyme substrate complex, thereby they inhibits its breakdown to the production of product. However, the EIS complex does not continue to form product, and non-competitive inhibitor lower the concentration of functional enzyme. Non-competitive inhibition cannot be overcome by increasing the substrate concentration. That's the reason, because no as such competition between the substrate and the inhibitor. So you better understand from this particular diagram. This is the substrate and this is the actual site of binding to the enzyme, and this is the inhibitor. So instead of binding at the actual substrate site, it can bind at the site, at another site, and that's how it is arresting the synthesis of product. Either it can bind to the this ES complex or it can bind to the enzyme. Both become impaired to lead to the production of product. So examples of non-competitive inhibitors are like ethanol or certain narcotic drugs, or are non-competitive inhibitor of acid phosphatase. Then trypsin inhibitor occurs in soybean, and raw egg white inhibits the activity of trypsin. Then Ascaris parasite contains the pepsin and trypsin inhibitors, and they inhibit the action of pepsin and trypsin. Here we will see the uncompetitive inhibitor. So initial was the non-competitive. Now this is the uncompetitive inhibitor. So these inhibitors can bind only to the enzyme substrate complex, not to the singly enzyme. It does not have affinity for the free enzyme, and the enzyme substrate inhibitor complex ESI does not continue to form any product. So uncompetitive inhibition cannot be overcome by addition of more substrate. So consequently, what the changes we will observe into the double reciprocal graph. So the Vmax cannot be attained, and even at higher substrate concentration. So this is for non-competitive inhibition, which already I have discussed. The Km value unchanged. Here the Km value unchanged while the Vmax is reduced. The Vmax is lowered. In case of non-competitive inhibitor, I forgot to discuss this. And now we will see for uncompetitive inhibitor what will be the scenario in the line we were plot. So this is the non-competitive inhibitor. First thing, it is binding. This is the inhibitor which is binding to the ES complex, not to the enzyme alone. So now this has arrested the transformation of substrate to product. But now because this system is locked. So what the changes you will observe in the double reciprocal graph? Uncompetitive inhibitors decrease both Vmax and Km. So this is the Km, it is decreased from here to here, and also the Vmax is also decreased, right? From here to, like, it is raising, mean it is Vmax, Vmax is the reciprocal value. So both Vmax and Km are reduced. Then examples are herbicide, glyphosate, known as the Roundup, is uncompetitive inhibitor of enzyme in the biosynthetic pathway of aromatic amino acids in the bacteria, then non-toxic in animals because they lack this enzyme. Now, an irreversible inhibitor bind with the enzyme tightly covalently. Earlier you have seen the binding is through not the covalent bond but the non-covalent interactions and here it forms an stable complex. Now, irreversible inhibitor cannot be released by dilution or dialysis or simply by increasing the concentration of a substrate. So, in terms of enzyme kinetics, the effect of irreversible inhibitor is like that of a reversible non-competitive inhibitor, resulting in decreased Vmax but having no effect on Km. So this kind of graph is being obtained. There is no change in Km while the Vmax is decreased. Now, alloesteric enzyme is a regulatory enzyme. The term alloesteric derived from the Greek word allo means other than the actual binding site. So, allosteric enzyme are those having other sites in addition to the active site of the binding of modulator. Allosteric enzymes may be inhibited or stimulated by modulator. The modulators that inhibit the enzyme activity are termed as negative modulators, whereas those that increase the enzyme activity are known as positive modulators. So we will see here, this is the enzyme. This is the catalytic site or active site. This is the alloesteric or regulatory site. So these alloesteric site can be both like the modulatory substance can be the positive modulator or negative. If it is a positive modulator, so bonding of this will facilitate the binding of substrate to the enzyme. Thereby, it will facilitate the formation of product. While the negative allosteric regulators, what it will do once it binds, so leading to some conformational change, which inhibits the binding of substrate to the enzyme. Thereby, there is no enzymatic reactions and thereby no production of product. Here, one important example of this allosteric inhibition is uh, in case of synthesis of heme. So reaction starts from glycine and succinyl coenzyme A under the influence of the enzyme alpha amino levulinic acid synthase. And this is the first enzyme in the synthetic pathway of the heme, and it converts up into the alpha amino levulinic acid. Further, after the cascades of reaction, after so many steps, converts up into the heme. Heme, once it is being formed in appropriate quantities, through allosteric feedback inhibition, it will inhibit the activity of this enzyme delta uh, synthase. So this is the example of negative allosteric regulation. Thank you.